Good morning, everybody, and welcome to the Dairy Research Conference and this session on dairy health. I'm delighted to welcome a host of speakers this morning, and I'd just like to run through a couple of housekeeping points with you. So all attendees are on mute at the moment. Please submit any questions that you have throughout the sessions. We will be having a dedicated question and answer session at the end. So we will take all questions at the end. We won't be answering questions in between the talks. We need to finish at 12:30 sharp, and the recording will be made available after the event. We will also try and answer any unanswered questions by email at the end. And there will be a short survey that we would just be very grateful if you could complete at the end. So my name is Mandy Neville. I'm head of AHDB Animal Health and Welfare, and I'm delighted to be chairing this session this morning. The first talk that we're going to have is from Matteo Crotta and Stephen van Winden. And Stephen is going to speak first, and I will hand over to him in a second. His talk is called The Relationship Between Cow Status, Transition Management and Johne's Disease Risk, Results of a Lifelong Cohort Study. So I will ask the other presenters to put themselves off camera, and I'll put myself on mute, and I will hand over to Stephen and Matteo. Thank you very much.
Thank you, Mandy. Yeah, so the relationship between the cow status and the risk of Johne's disease. And I think it's really important to acknowledge the fact that we were able to prolong a study that we did 5 years before this started, because that allowed us to follow through the cohort in its full length. And I'm Stephen van Winden. I work for the Royal Veterinary College, and I'm an associate professor in population medicine. So if we look at the next slide, we can even the following slide. We can have a little chat about the study. And so the study design is a cohort study, and it is a prospective study. So basically, we're going to start today, and we're going to follow the cows through as they progress in life. And of course, we all know that Johne's disease is a slow progressing disease, so therefore we need to have a long breath in this. And if we look at the next slide, we can see how schematically that is being presented. So we started off with 600 female calves born on 6 commercial dairy units. And these commercial dairy units were testing routinely for Johne's disease, as we all know them commercially available. So we've got lots of information on these individual test results, but also historically of their dams that these female calves are being born from. So the bits that we recorded is on the left-hand side of this slide, and they relate to the status of the dam, but also on the environmental conditions that these calves were being born into. So because we know there's a maternal part, call it the vertical transmission, and we've got a more horizontal part which relates to risk factors that we identified, and these are the bits that we recorded, and they'll feature again towards the end in analysis of the data. Apologies. So it all started cows being born in 2012. So this is nearly 10 years ago when the study started, and we continued recording their data up to March this year. So it's a long-term study, and that's a thing that we probably would need because there's otherwise an issue of having not enough cases in our study design. So if we look at the next slide, we can see how many of these calves, 440 in total, were grown into from the 600 baby calves into adult calving cows. And in this slide, the table shows how many in total were enrolled, but also how many actually made it to become a mature dairy cow. And you can see that per farm, there's quite a bit of variation. So some will go up to 90%, the lowest was nearly 2/3. So these cows did not make it into the production lives and were culled for other reasons. So, and I think it's really important to realize that these farm factors that play into that are being controlled for in analysis, which we can see in the next slide. Sorry, the next slide first shows us the number of positive cases that we see it develop over time. So that we start off on the left in 2014. So these cows are born in June, give it 2 years before they are being productive cows. And then subsequently the first test results will come in. And as the test results come in, we will taint cows as positive cows. And these cows are qualified as positive as at a 30% SP ratio. That would be equating to amber cows. So that is one single positive result is a flag for these cows to become JD positive in this study. So these are not the same as our red cows, but it's important distinction for us to make that we include all these cows. So, and if we then look at the number of cows when they test positive, we can see also quite a discrepancy between herds. So the first dots are only a few because the disease is still incubating. 2015 to 2017 is when we got the biggest hit rate of positive cows, and then subsequently that tails off. Part of that is because there's fewer cows on those herds still alive, and therefore there's fewer candidates of cows develop Johne's disease, but also, of course, Johne's disease typically will present itself not very late in life, typically lactation 1 to 3, maybe 4, and then it starts to tail off, and that is also depicted in this slide. But as you can see, both on the rearing success but also on the JD conversion, there is a variation per farm, and the next slide will talk about how we try to untangle that spread of that disease, and these farm factors are included in that analysis. So in the bottom line it says it is a stratified design, so therefore we account for the variation that are systemic on those farms. And it's really important for us to consider that, because otherwise it will be difficult for us to generalize any of the outcomes. So as mentioned, an SP ratio of 30% is sufficient for them to be flagged as a positive cow, and then subsequently we look at the risk factors that we collected in the first bits, in the first part of the slide, for us to be able to untangle which factors contributed most to that spread of that disease. And if we describe the different routes, we basically have the maternal, call it the vertical route, where there's either the in utero, so when the calf is still incubating in the womb, is playing a role, but also the colostrum route plays a role, and we can untangle that to a degree. But of course, we do need to know the dam status as well as the source of the colostrum. And then subsequently, alongside, but in some cases separate to that, we also have the fecal-oral route. So that's basically when there's dung contamination of these calves going into these calves, and therefore subsequently giving them a sufficient dose to convert into Johne's disease. And factors for that, of course, include colostrum, but also environmental factors such as the cleanliness of the yard, as well as how long they spent in that calving pen, whether it's longer or whether it's shorter. And these factors are subsequently described in a graph in the next slide, and I think that's really informative for us to have a look to basically unravel the routes that we have available for these calves to become infected. So on the left-hand side, we've got a box, which is the farm, and that is up to now a black box, and then slowly but surely we start to unravel that black box into the routes of infection. So the typical one, dam, is the mother is the A, and then subsequently it goes to a positive Y, that is the calf, the offspring of that cow. And of course the in utero route is a straight line, but also there's a route that goes via the colostrum, and which goes via the top bubble, so that's the let me see what number, that's bubble C, sorry. And then of course we also have the bottom route, which is the environmental route, which we try to untangle in two different routes. So we've got the D route, which is directly from mum to calf, but also there's the E route, which goes from other cows onto the calf to become infected and to receive the infectious dose. And these are the different routes that we then subsequently try to untangle in the analysis, which is described in the next slide, where we basically allow only cows in the model. For instance, in the first one, where we're trying to untangle the relationship of dam colostrum and dam status in utero by only taking the cows, taking the calves that have received colostrum from negative cows only. So that is our way of trying to identify these different routes in a separate way. And then, of course, also we've got the other routes that we want to account for, and it's really difficult to untangle the in utero route versus the environmental route, but we are able to identify the environmental route from others. So these are the different routes that we tried and successfully, as Matteo will talk about later on, identify the routes and how much they contribute to the chance of infection of Johne's disease onto those calves. Which brings us to, I think, introduce Matteo, who will take over the next part of the presentation about what did this inform us. Yeah, so Matteo, the floor is yours.
Thank you very much, Stephen, and good morning, everyone. My name is Matteo Crotta, and I work as a postdoctoral researcher at the RDC. I will be presenting you the results of our analysis, starting with the first slide, with the model where we explore the association between the status of the dam and that of the cows. So the first model was evaluated not considering whether the dam had already shown evidence of seroconversion at the time of calving. So in this case, a positive dam is a positive dam, regardless of whether seroconversion was detected before or after calving. Next slide, please. As we see, so what we found in this case is that there is a positive association in that cows born from a positive dam are about 2 times at higher risk of JD infection. Now, considering the strong correlation between being born from a positive dam and having received the colostrum from a positive dam, we have also repeated this model, but this time only considering cows that had colostrum from a negative dam. And the results actually reinforced the previous finding in that cows born from positive dams were at more than 2 times higher risk of JD infection. From here, we have used this data set to check whether the risk of infection is different between cows born from the dams that the farmer knew were positive at the time of calving and cows born from dams that shown evidence of seroconversion only after calving. And indeed, what we found is that the higher risk is actually coming from the cows that the farmer didn't know they were positive at the time of calving. So this means that yes, the risk is higher in cows born from positive dams, but this higher risk is unrelated to the actual detection of the antibodies at the time of calving. As we see in the next slide, for the cows that were born from negative dams, we have explored the environmental route of infection under the hypothesis that spending a long time in a dirty yard represents a risk factor for the fecal-oral route of infection. And indeed, what we have seen in the next slide is that cows born from negative dams, but spending a long time in a dirty yard, have about 4 times higher risk of infection as compared to the reference group, which are cows that spent a short time in a dirty yard or cows that were kept in a clean environment. Now, using this data set, we have also tried to compare, as shown in the next slide, the 2 different routes of infection, the in utero and environmental route of infection, in terms of the time to detection. So if we look at this Kaplan-Meier curve, survival curves, for cows that very likely got the infection from the dam, which is the line in red, and cows that very likely got the infection from the environment, which is the line in green, we see how at the median we have a difference of about 7 months. So this means that half of the infected cows that presumably got the infection from the environment were detected 7 months later than half of the cows that got infection from the dam. Now, as you can see, the confidence intervals are quite a lot of overlapping, and this is probably due to the low power for this type of analysis. So this means that even if the cure seems to suggest a principle of two different trends, we could not really prove it. Now, as we see in the next slide, for the cows in the cohort, we also had production and fertility data and somatic cell count, but we could not find a systematic difference in these parameters between the JD positive and JD negative cows in the cohort. So in the conclusion, in the next slide, there are a couple of bullet points. In conclusion, we have evidence to say that cows from infected dams are at higher risk of JD infection, but this risk is unrelated to the actual presence of antibodies in the dam at the time of calving. For cows born from negative dams, we have evidence suggesting that the cleanliness of the yard in combination with the time spent in the yard is a risk factor for infection. When comparing the time to detection, again, the trend suggests that there would be merit for further consideration, because although we could not prove it, we have some signals from which it seems that cows of which infection can be attributed to the environment are detected later as compared to cows infected from the in utero route of infection. And again, no systematic differences were found between the JD positive and JD negative animals when looking at production and fertility data. But in this case, as for the time to detection, we could not really rule out that this is because of the relatively few infected animals in the dataset, and therefore the power of the analysis. So with that, I have concluded, and I really thank you for your attention, and the AHDB for the opportunity of putting together such a, I would say, unique data set. Thank you very much.
Thank you very much, Matteo and Stephen. That was a very clear presentation, and some, just what a fascinating study to have been going on for so long. It's rare as a researcher that you get the opportunity to work on such a study, and I think it's absolutely fantastic that we're seeing the highlights of that study coming through now. Okay, so if we move on to our second speaker, we will have two speakers again in the second session. So we have Erica Nunney and Javier Guitan, and I think Javier is going to speak first, and then Erica will follow in a similar style to Stephen and Matteo. And they will talk about the effect of tuberculin skin testing on Johne's disease test results, evidence of distinct effects in cows with and without Johne's disease. So if I hand over to Javier now. Thank you very much, Javier.
Thank you, Mandy. And I echo what Matteo have just said. Thank you for the opportunity to present some of our results. Now, Erica and I are going to present a parallel activity that we did as part of our work on Johne's disease. Although it was done as part of the second research partnership, in fact, it started with the first partnership. Next slide, please. During the first partnership, we worked on trying to integrate the information that we accumulate on individual cows by repeatedly testing them in a way that is a little bit different to the traffic light system that has been used. There is a lot of information that is collected and accumulated as cows are tested during their lives. And we wanted to make use of the actual values and integrate them with additional information, such as the age when these results were obtained. And what we see in the chart on the right is On the vertical axis, the probability that the cow is infected and how it relates to the traffic light system, whether the cow is red, green, or yellow. And as expected, we can see that they are very closely related, but not perfectly. So we have, for example, some red cows that may have a lower probability of being infected according to our algorithm than some yellow or even green cows. So this is the idea that we've been trying to pursue, trying to make a more integrated interpretation of these test results. Next slide, please. And as part of our work, we had invaluable discussions with stakeholders. Along the project, we shared our results with a number of people during stakeholder meetings, and then they provided us with extremely useful guidance. And one of the issues that they raised was, what is the impact of TB testing? How does TB testing affect JD test results? Can you look at that in your project? And as a result, we added 2 objectives as part of our project. Next slide, please. To assess the effect of the time since TB testing on the JD test values, and to do this for both cows that were likely to be infected and cows that were likely not to be infected. Does it have the same effect or not? And to estimate the impact of the proximity to TB testing on the sensitivity and specificity of the JD test. So what is the performance of the JD test if done close to TB testing? Next slide, please. I think it's the previous one. Yeah. So the study, contrary to the study that Matteo and Stephen have just presented, which was based on generating our own data in the field, now we made use of existing data. We use data from the 2 main milk recording companies on JD tests that we combined with TB test data from APHA. Next slide, please. And we ended up with a large data set of around 250,000 JD test results from almost 100,000 cows that at some point were tested within 3 months of a TB skin test. Next slide. Now, our approach was trying to compare the time until a JD positive result for cows that retrospectively we know that are likely to be infected because we have the whole history of testing, but at the time of that specific test were not yet known to be infected, and cows that we think were not infected. To compare the pattern of SP ratios. And what are these SP ratios? When we get the JD test result, positive or negative, is based on a value. And this value is the SP ratio. Basically, the SP ratio is the way in which the JD test result in milk is summarized, is this number. And to assess, we assessed the sensitivity and specificity of the test when these tests are done at different times after TB testing. Next slide, please. The classification of cows as likely to be infected or not was based on the herdwise program definition, a definition that we are all familiar with. And for the JD milk ELISA results, we use also the usual cutoff of 0.3. And with this, I'm going to leave you with my colleague Erika, who will go through the next slides and will go in more details over the methods and the results. Thank you.
Okay, so to examine the first objective, examining how time between time of TB testing and your time of JD testing affects the real test results of the JD test for MAP infected and non-infected, we did 2 different approaches. So if we go to the next slide, so the first approach was a Cox proportional hazard model. So that's examining time till your event of interest. And for us, the event of interest was a JD positive test result. And the second approach was to use a generalized additive mix model. This is essentially a linear model, but it uses smooth functions, which allows for flexibility in the model, because instead of just assuming that the relationship is linear, you're able to examine non-linear relationships. So if we go to the next slide. So the first approach, it's called a nested frailty model we used because to take into account that observations in the data set are not all independent. So each cow has multiple JD observations and cows belong to different herds. So in order to take into account of that, essentially added a random effect terms for each of those. And so we examined JD tests that occurred within 90 days of TB testing, and we split this variable into 4 different groups. So 0 to 14 days, which is JD tests that occurred within 14 days of TB testing, and so on. And these groups were compared against observations that occurred 61 to 90 days post TB testing. And so if we go to the next slide, we then see the results. So looking at cows that we've classified as infected, we can see that the results suggest that there's a high probability of testing positive for JD among those that we believe to be infected when testing close to TB testing. So it has a ratio of 1.44, and then the trend slowly decreases for infected as time from TB increases. And so if we go to the next slide, this is the same table, but just highlighting that in cows that were classified as non-infected, the hazard ratio is highest when JD testing occurs 15 to 28 days post TB. So this sort of suggests that there may be a difference among cows that have been classified as infected and non-infected. See that got a higher hazard ratio for non-infected, 15 to 28 days post TB, whereas in infected it's highest closer to TB. So to examine this relationship a bit further, if we go to the next slide, we looked, so using generalized additive mix model, instead of looking at time from TB testing as a classical variable, looked at it on a continuous scale, and instead of looking at our outcome being a JD positive test result, we looked at how this affects the actual SP ratio value of the JD test. And so if we go to the next slide, so from these results, so you can see on the x-axis is the time of JD testing in proximity to TB testing, and on the y-axis it is the SP ratio from the milk ELISA. And so the black line represents the model for the relationship between time from TB testing and the SP ratio value for infected cows that we've classified as infected. And the black dotted line represents the relationship for cows that we've classified as non-infected. So you can see that for cows that were classified as infected, they have a higher, it looks like they have a higher SP ratio closer to TB testing, and then this slowly decreases as you test further away from TB testing. Whereas for non-infected, they have a peak of the SP ratio around like 14-15 days post TB. So it's not an immediate reaction to the TB test. So you don't see that immediate increase like you do for MAP infected cows as you do for non-infected. And this is what sort of saw in the survival analysis, where the hazard ratio was highest 14 to 28 days post TB for non-infected, whereas for infected it was higher, closer to TB. And so if we go to the next slide, so to see how this impacts the actual characteristics of the test, so the sensitivity and specificity. in order to meet this second objective, if we go to the next slide, estimating sensitivity and specificity. So randomly selected one observation from each cow and repeated it lots of times to estimate the mean sensitivity and specificity of the test. And so if we go to the next slide, we can see that, so estimated sensitivity, so the mean sensitivity overall, highest when testing close to TB. So the proportion of diseased individuals that test positive is much higher when testing 1 to 14 days post TB. And this is sort of seen, if we go to the next slide, when you look at the GAM, you can see that for cows that were classified as infected, they're more likely to have a high SP ratio and also more likely to test positive when closer to TB. So that reflects why the sensitivity is potentially higher when closer to TB testing. But soon non-infected do eventually have get an increase in the SP ratio value, as you can see from the GAM, which could lead to false positives. So if we go to the next slide, you can see that it drops the specificity when testing 15 to 28 days post TB, and it's higher when testing further away from TB. And so you can see that trend there. And then, so moving on to the next slide. So then we estimated positive predictive values, negative predictive values, and the overall test accuracy. And so in order to do this, we simulated, made a simulation using binomial probability model. So using the mean sensitivity and specificity for each of the time periods from TB testing, and different prevalence levels. We estimated the number of true positive and true negative animals. And so if we go to the next slide. So from the results, so looking at positive predictive value, so the probability that an animal is truly diseased, truly infected, given that they've had a positive test result, it's lowest when in that time span between 15 to 28 days, which follows that trend where the specificity drops then as well, because you get potentially some false positives from cows that aren't actually truly infected with the disease. They're actually negative. And you can see that it's highest when testing 61 to 90 days post TB, and at both prevalence levels the same trend is seen. And if we then go to the next slide, you can see that the negative predictive values, so it's highest when testing close to TB, and that reflects what was also seen in the sensitivity. It's highest then because you get that increase of infected animals are more likely to test positive / have a higher SP ratio when close to TB testing. And then that slowly decreases as time from TB increases. And then if we go to the next slide, so here we've estimated overall accuracy of the test, so the proportion of test results that are actually correct. And there's not much variation in the accuracy between the different intervals, but it does drop for each of the different prevalences at that time, 15 to 28 days post TB testing. But yeah, you see much more of a change in the accuracy as prevalence increases rather than over the different intervals. And then go to the next slide. So in conclusion, the results suggest that there is a difference between cows that have been classified as infected and non-infected, and how TB testing affects their JD test results. And what you see is that increase in sensitivity close to TB testing, but also that drop in specificity, a bit like a lag after TB testing. These results, however, they're not published. We're still looking in detail to see if this relationship is, truly the true relationship. But yeah, I'm now going to pass you on to Javier again to talk about the next steps.
Thank you. So I think just to summarize, in the previous presentation, Matteo and Stephen refer, for example, to the role of the relationship between dam status and the probability that the cow is infected or not. Erika has just explained the impact of the time since TB testing. So it may be of interest to take into account when we get a JD test result, when it was obtained, not only in terms of the age of the cow, but also how far from a TB test. And ideally, when making the decisions that are made at the level of the farm, taking into consideration whether the dam was infected or not. And this is what we've done as part of the research partnership. We have created a model, a framework that allows us to combine all the different results of an individual animal based on when they were obtained, at what age of the cow, and how far from TB test, the status of the dam, whether the dam was infected or not, and based on that, producing an output which is a probability of infection. And we've been fortunate to receive some additional funding for the coming months to work on two things. One is validating this tool. We are going to do some field work by testing in the field in fecal samples and also in blood for the presence of MAP in cows with different probability according to the algorithm. And we will be working with the milk recording companies in trying to implement the algorithm so that it is available for farmers when they test their animals, excuse me, for JD. The beauty is that this probability is updated. As we accumulate more information, the probability is updated. Next slide, please. Well, just again, acknowledging AHDB for their support and a large number of people who have been involved and made the study possible. Thank you.
Thank you very much, Javier and Erika. That again is a fascinating piece of work that I know jointly with Stephen and Matteo is yielding some really fascinating results and outlining some of the complexities of the diseases and the links between TB and Johne. So the interpretation of results, I should say, from those two diseases. So absolutely fascinating. Thank you very much, both of you. We will move on to our third speaker, who is Professor James Breen and James is going to talk to us. Let me just get my title correct. Great. James is going to talk to us about the mastitis pattern analysis tool. James is one of a group of researchers at the University of Nottingham and will update us on his work through the research partnership. Thank you, James.
Good morning, Mandy. Good morning, everyone. Hopefully you can see my title slide there and hear me okay. Thank you very much for the opportunity to speak. As Mandy said, I'm part of a research group at University of Nottingham, and I'm going to give you a short talk on how we've developed a pattern analysis tool to use to prevent new infections in dairy herds and to understand infection patterns in dairy herds. Why is mastitis important? Very briefly, by way of introduction, we know obviously there's great emphasis on antimicrobial use and therefore great emphasis on preventing infection to therefore avoid the need to use antibiotics. Mastitis is still important for a range of reasons, both financial, welfare, of course, because it still remains quite prevalent within our national herd, and as well as milk quality, sustainability issues. The antimicrobial agenda and prudent use was really put front and centre in recent years. It also provides a great opportunity to engage with our dairy clients with herd health and actually do herd health and preventative medicine. And there's been a lot talked about in terms of dairy cow welfare and the sustainability agenda. But really, we came at this a few years ago off the back of the O'Neill report, as I'm sure you'll remember, and the importance of a review of antimicrobial resistance. And that drove then the Rumour Targets Task Force in 2017. And the dairy sector then discussed those, if you like, targets to reduce antimicrobial use within our sector. And when you look at the targets that we were asked to review and comply with, 4 of them put mastitis really front and center once again, because they focused on intramammary use and dry cow therapy and such like. So this was our sort of thoughts around, well, of course, mastitis is important in terms of antimicrobial use if we just consider this and not the financial welfare aspects. We know it's important because lactating cow intramammary use and dry cow intramammary use take up a lot of daily doses and courses. We also know that injectable antibiotics are still often used, and unfortunately still often used quite inappropriately for mastitis therapeutics, and they take up a lot of your milligram use. And of course, in recent years, less so now, perhaps, the critically important antibiotics were used to treat mastitis, particularly severe mastitis events. And we thought, well, of course, if you review mastitis control through the lens of reducing antibiotic use, how do you reduce antibiotic use through improved mastitis control? There's been a lot of focus on not treating some clinical cases, and that's the on-farm culture story and such like, and we haven't got time for that this morning. That remains quite controversial in some ways. We can clearly modify approaches to treatment, and a great example has been the adoption of the selective approach to antibiotic dry cow therapy in the last 10 years or so. But we as a group really come at it from the point of view of avoiding the need to treat. So if we can reduce the occurrence of new cases, new infections, maintain mastitis control, well then of course we don't need to think about treating with antibiotic at all. That's been well publicised through the AHDB Dairy Mastitis Control Plan. And we came at this from the point of view of, well, that's been widely used and widely available for a long time now. But could we provide something that actually people could use that was perhaps easier to use and more widely available than the actual plan, and then dip into the mastitis control plan as necessary? So if we go back to mastitis control very briefly and think about how you control new infections, of course, mastitis control is not simple, because farms and their pathogen profiles and their patterns of infection are very different. And this was the mainstay of what underpinned the mastitis control plan all those years ago, that you couldn't do a one-size-fits-all approach. It just doesn't work. We have to consider where new infections come from in our herds and work with herds on a herd-by-herd basis. People do still focus on infected cows as a source of new infection to other cows in the herd. This is the so-called contagious mastitis transmission model. We do assume this happens a lot still, and we focus our efforts on treatment and or culling because we assume infected cows are transmitting infection to uninfected herd mates. It can be quite reactive. It tends to rely on antibiotic treatment, which of course is unsustainable. And we have to ask the question, how many of our herds actually have contagious mastitis patterns in terms of the predominant pattern is contagious? Because also the other route clearly is new infections from the environment. And this is more complicated. This can be more difficult for us to understand because we're thinking about bedding, water, buildings, paddocks, loafing space, summer, winter. It can be a real challenge to control environmental infections. But modern mastitis control needs this distinction. We have to first of all understand what the farm-specific diagnosis, if you like, is. We can't just go charging in and putting in place interventions on the assumption that things are happening. We have to have this diagnosis of the pattern, because then we have to relate that to areas of risk on farm and then make changes that target these specific areas. No point talking to our producers about cubicle bed management, for example, if 75% of the new infection pressure is coming out of the dry period. We're in the wrong area. So we have all these things going on with lactating cows at pasture, dry cows in loose yards, milking cows in cubicles. How do we know which areas to focus on? Is it the parlour? Is it teat preparation? And of course, by looking at the data and coming to an informed decision about the pattern, we then say, right, we need to focus on environmental management of milking cow cubicles in the winter, for example. How do we do that? We need individual cow somatic cell count data. I need to know that cow 2240 there highlighted on screen has moved from a very low cell count, 20,000 in the May, to a very high cell count, 3.1 million in the June, because this would be a new infection. And I need to know how many cows are like her, and if this is a pattern that tends to happen at this time of year. We also need the clinical mastitis event data. And we need to know, for example, that clinical cases that are occurring within 30 days after the calving event, coloured blue here on the graphic on screen, well, these are very likely to be of dry period infection origin. And therefore, does our herd of interest experience a lot of mastitis events within 30 days after calving? These things are patterns, and these are what's the sort of data we would look at and come to a decision about the predominant pattern in the herd. But could we go one step further and automate this? We know this is a barrier. Achieving a herd mastitis diagnosis, if you like, can be a real barrier out there to mastitis control on farm. It requires a bit of thought and a bit of time to look at this data in a structured way to come to a decision. What about developing some sort of pattern analysis tool that producers, vets, advisors could use that would accept data about cell count and clinical mastitis events and tell us where and when new infections are probably occurring, so people could get on with the prevention and not wait until events or infections had happened and then be having discussions about treatment. So briefly then on to the mastitis pattern analysis tool. This is what we're developing, how it works in practice. The whole basis of the research was, could we make this process of assessing herd data very easy and very quick? That if we could mine the herd data from the milk recording organization or on-farm software, this database would contain all the individual cell count data, contain all the clinical mastitis events. It could be sucked into this analytic tool and then hit a button, and 3 seconds later we've got an output of the predominant mastitis pattern and therefore an idea of where we need to focus. Developing the pool was essentially around taking well-known metrics using individual cow cell count data, for example, a dry period new infection rate, proportion of cows that are below 200,000 prior to drying off, that are above 200,000 at first test day after calving. Take the clinical mastitis event data recorded by producers into a database, and again use well understood metrics that we've taken for years with the mastitis control plan, for example, the proportion of clinical mastitis cases in the first 30 days, and use this to look back over periods of time and compare it to an intervention level, a target, if you like. We would also need information about heifers at their first calving event. What proportion of heifers post milk recording somatic cell count results above 200,000 at their first test? What proportion of heifers are reported with clinical mastitis within 30 days of their first calving? Those sorts of things would be looked at for a 3-month period, for the 3 months prior to that, for the 3 months prior to that, and so on and so on. So we'd had 18 months worth of these metrics. So we could then also compare seasons, and we could see the presence of seasonality in these metrics. Really important to highlight environmental mastitis patterns, for example, if there's clear differences between periods in the winter and periods in the summer, turnout, housing, and so on. So the pattern analysis tool began life in this way, because if you know the herd pattern, you can then predict, of course, what the predominant infection pattern is, and we can look ahead. Well, we know we have periods where new infection rates in the milking cows rise through the winter, so therefore we need to focus on this area now as we go into winter housing 2021-22. We then tested the pattern analysis tool, so we had a sort of a training phase. We went back to the original mastitis control plan herds, where we have a huge amount of information. We took 50 herds with a known diagnosis, if you like, so we're using a diagnosis made by specialist vets. Back in the day, we've got that information. We could give that information to the pattern tool and see if it agreed with the diagnosis made at that time. We then had a testing phase where we took another 50 herds and we had agreement in 90% of herds. So we're looking for whether the pattern tool can agree on a contagious or an environmental pattern, and whether it agrees on a predominantly dry period infection origin or predominantly lactating period infection origin pattern. So how it works in practice is, of course, we need the milk recording data. So we go to the milk recording organization and we're looking for a CDL file, a common data layer. So the farmer or the vet or the advisor would download this herd milk recording data from CIS, NMR, QMMS would provide this common data layer, and this would contain all the cow health and performance events, calving, clinical mastitis. It would also contain all the individual milk recording data. That CDL file is then converted using free software. This free software is downloaded from the AHDB Quarter Pro webpage. This has been provided by QMMS and Summit for use by the industry. There's a YouTube video there designed for you to just see how it works, but essentially it calculates all those metrics I've just described, new infections, new cases, and drops it out in CSV format. If you find that your producer is not reporting clinical mastitis events to the milk recording organization, and unfortunately this can be quite common, you have the opportunity to merge a spreadsheet of clinical mastitis event dates and cow IDs into the tool at this point. So you can merge clinical mastitis event data that you've captured from the diary or on-farm software, for example, and then we can generate the output file. We might just have to think about the calving pattern. For herds which are block calving, we might have to make sure we're looking at 3-month periods that align to when cows are calving into the herd. For all year round calving herds, that matters less. We might just need to think about winter housing, summer grazing, turnout, and so on. But we're thinking about 18 months worth of data. We then generate this file and then import that file into the pattern tool, the second of our free softwares available from the AHDB Quarter Pro webpage. Again, another YouTube video designed to show you how to use it, but very easy to use. We import the file into the pattern tool. That takes about a second or two. All the metrics are there. We hit the next button to assess the quality of the data. For example, do we have enough milk recordings? Do we have enough clinical mastitis events? We hit the next button and we can then have the actual pattern output. And this is on a traffic light system. And this looks at the predominant pattern in the last 3 months, current, the previous 12 months, recent. And in this example here, we can see that the predominant current issue is one of environmental infections in lactation. The pattern tool takes the metrics we've calculated and gives a score, if you like, in inverted commas, out of 100. So points win prizes effectively. Seasonality, high new infection rates in the dry period, and so on will give you a higher score for a dry period environmental pattern. A high prevalence of infection, very poor cure rates across the dry period, no seasonal patterns will give you a very high score in the contagious bracket. There's more information available within the term, including a glossary, terms and definitions, and some initial pointers. But we wanted to go further and then provide the resource material. So once you have your mastitis infection pattern, you'd say, well, what next? If my pattern is one of environmental mastitis in lactation, can I go to the AHDB Quarter Pro webpage and download a series of little fact sheets in that area? Fact sheets that give us some of the headline findings from the original Mastitis Control Plan work, so we can sit down with the producer and the advisor and start thinking about ideas within that area, within that predominant infection pattern, that we can start to make interventions. Of course, no point using that resource if the pattern tool tells you the predominant infection pattern is one of environmental infections during the dry period. You need the dry period environmental pattern resource, and so on with contagious mastitis. So we target our interventions to the predominant infection pattern at that time. We therefore optimize mastitis control. So within the Quarter Pro scheme, predict P, react with resources R, optimize Quarter Pro. And we do this to make sure farmer and advisors are working in the right area. Like I said, no point talking about lactating cow cubicle management if 3/4 of our mastitis infections are arising from the dry cow environment. We need to target our time and resources to the right area. And of course, predicting the pattern in this way means we can stay focused. Roughly every 3 months, we can convert the mastitis and cell count data, sit down with our producer, check that the pattern is changing. It might be changing. We might have seen the impact of interventions we've made. The pattern has now changed and we need to move on accordingly. Predict the most important source of infection on the farm and make sure we're advising interventions around management, environment, milking routines, whatever, but that has to be within the right area. So in summary, the pattern analysis tool is available using this web link. It's free to download, and we're trying to get producers, vets and advisors to remember that when working with herd mastitis control, we need to understand when infections are predominantly happening. Is this during the dry period? Is this in lactation? Where infections are predominantly coming from? Is this from infected cows, or is this predominantly from the environment? Because if we know this, we can then identify key risk factors on farm, use the resource material to work out what can be done to reduce these risks and make changes. Find the mastitis pattern tool online. We have also now embedded the pattern tool and the information around the pattern tool with the Quarter Pro scheme, embedded this in the British Cattle Veterinary Association CPD, so advisors, vets can take this series of training modules and become Quarter Pro advisors and go on and use the pattern tool out there. I'd like to acknowledge the group. Obviously, I'm just part of a team here, so Professor Andrew Bradley, Professor Martin Green, the original idea, Derek Armstrong at AHDB, my colleagues Pete, Chris, Catherine and Bobby, and all the testing and design by QMS and Summit, University of Nottingham, Mastitis Control Plan Steering Group, and to name check Rachel Hayton, Ian Onstad and Tracy Towers, of particular use to us testing this and going out in front of farmers. Thank you very much indeed. Take questions later on, and I'll hand back to Mandy now.
Thank you very much, James. That's a fascinating oversight and just illustrates the power of data, I think, enabling you to target some prevention in that all-important responsible antibiotic use arena. So could I just encourage participants to put some questions in the chat? We'll take them at the end. And any that we can't answer, we will email. But do think about putting a question in the chat if you have one. I'd like to move on to our final speaker today, Hayley Crosby-Durrani from the University of Liverpool. And Hayley's going to give us an update on bovine ischemic teak necrosis. Thanks very much, Hayley. Over to you.
Thanks, Mandy, and I'd just like to say thank you for allowing me to talk today. Teak necrosis research is really in its infancy, so being able to talk here and increase awareness of the disease is great. So thank you. Hi, I'm Hayley. I'm going to give you a bit of an update on teak necrosis. I'm a veterinary pathologist and a PhD student at the University of Liverpool. So what is teak necrosis, or ITN, or ischemic teak necrosis? Well, it seems to be an emerging disease. And how does it present? It looks like a well-demarcated black, red sore on the teat, usually on the inside of the teat, midway to the joint where the teat and the udder meet. We seem to have a few different presentations. So there's presentation like in the first picture where it's really dry. There's one where the teat then becomes really crusty, like in the second picture, and then another one where the animal's actually lost her teat. Either this is due to the fact that the disease can be really quite itchy and the animal will constantly lick at her teats and self-traumatize herself, or due to the process of necrosis, the dying tissue itself. We are still investigating the full pathogenesis around that. There was a pilot study involving 12 animals that suggested that teak necrosis may have been linked to digital dermatitis and had similar bacteria present in these lesions. So we've had a look to try and see if that holds true. So if we move to the next slide, how have we gone about investigating this? Well, What have we done so far? So we've done extensive farmer interviews either on the phone or with farm visits. We've also had countless numbers of vets contact us, and we've discussed cases either over the phone, via email, or going to CPD events. With AHDB's help, we were able to put out a national farmer questionnaire to over 1,800 farmers, and we managed to get the information of that questionnaire, some of which I'll present later. And we've been able to collect samples of swabs of these lesions or tissue of the lesions and process to look at the bacteria present in some of those samples, and some of them we've been able to look at under the microscope. We've got more samples now, but I haven't managed to finish all the testing on them yet to present. We've also had a look at, done a shotgun metagenomic study, which basically looks at all the different types of DNA present in some of these tissue samples. So what did we find? Well, people have been reporting that this is widespread across the UK, so it's not across GB. So it's not just a problem in one area, say the southwest. It's a problem of scattered farms throughout GB. And from our national survey, we found that of the people who responded to that question, that just over half of the farmers have actually seen a case of teat necrosis, which was far higher than what we had anticipated to start with. And we found that there was no difference between year-round and block calving systems. Okay. And some farmers have other names for these lesions, such as digi of the udder, probably from the first pilot study, and teat licking. Okay, so if we just have a look at what was reported, the sort of signalment of the animals that were reported to have lesions. So we found that it seems to be a really first lactation heifer disease. So nearly half of all of our cases have been in the first lactation. And then we have some who come down in the second and third and fourth lactations, and very few older cows will come down with teat necrosis lesions. Very, very few as well with pre-lactation heifers. So it seems to be predominantly a first lactation problem. And whereabouts in the lactation are we having cows coming down with these lesions? Well, it seems to be early on. So most of the time it's within the first 30 days or the first 30 to 90 days of lactation. So when these animals are under the high stress of entering the dairy herd and the milking herd. So we need to really be careful and keep an eye on these first lactation heifers. And if we can move on, next slide. So as I said, it seems to be an emerging disease. One of our questions of our farmer survey was when the farmers who responded had first seen a case on their farm, so the first case that they'd seen. And you can see with the graph on the left that the number of farmers reporting the first case on the farm is increasing in the since the millennium, basically. The number of at 2018, there's only 2 farms there. The survey was posted in January, so the fact there was 2 farms in January is interesting. So that's why there's a big drop off between 2017 and 2018. So one of the things the survey hoped to try and find was potential areas to look at as to why some farms may have cases of teat necrosis while other farms don't. And we've performed a series of regression models to try and find potential risk factors to investigate further. And what we found was farmers that were reporting having cases of teat necrosis in the younger lactating animals tended to have more cases of udder cleft dermatitis in their older cows. So that makes you wonder, is there something going on there? And also they tended to have more problems with the teat health itself. So we tended to have teats that were drier and chapped on the farm, so that had cases of teat necrosis compared to the ones that didn't. So this is something that we need to have a look into in more detail in the future, is are these diseases affecting the skin of the udder and the overall general health, meaning that people are more likely to notice and that you've got teat lesions, or are they're truly linked to the disease development? So this is an area we need to investigate further. So as part of the study, other than the questionnaire, if we move on to the next slide, please, we had a look to see if we could try and find potential causes for this disease. So there was the pilot study by Simon Clegg and others back in 2016 that found that some of these teat lesions had the same bacteria present as what's been associated with digital dermatitis. So we've basically tried to hunt through our samples, looking for the DNA of them bacteria, and also looking using immunohistochemical methods at the tissue sections under the microscope to see if we can look for the bacteria there. And also the farm history, does the farm have digital dermatitis problem or not? So when we looked at the DNA specifically for these digitrypanomes, the bacteria that's associated with digital dermatitis, we found that less than a third actually had it present. So this doesn't correlate well with the pilot study, so maybe it's something else. So we also did the metagenomics on some of these teats and some control teats. So if we look at the image on the right hand side of this slide, you can see we've got M1 to M10. These are teat necrosis lesions. So this is our shotgun metagenomics that we've done, looking at all the different DNA present in the samples. So M1 to M10 are our teat lesions with teat necrosis, and M11 to M20 are our control animals and the control teats. So if you look towards the top of the image on the right hand side, you can see where you're comparing the controls bacterial populations. The closer to red the image is, the more similar the bacterial population is. So we can see that all our controls have quite a similar bacterial population present. But then when we look down towards the left bottom side of the image, this is when we're comparing our teat lesions to each other. We can see that there's a lot of differences between the different bacterial populations on each of the different teat lesions, which is where it's more blue and more white. So this makes us think, well, maybe it's not a single one cause that causes this. Maybe it's a dysbiosis, a problem with the bacterial population, a bit like you think of with probiotics for good gut health. Maybe we've got a problem with the bacterial population, and we've got bad bacteria overloading the good bacteria in these teat lesions rather than one bacteria causing the problem. So if we move on, because I can see I'm coming towards the end of my time. So what are the key things that we've found so far? So we found 3 different presentations. So we have the type 1 lesion where we've got the dry area of necrosis. We've got the type 2 where it's all crusty, and the type 3 where she's lost the teeth or it's been traumatized. We find that the first lactation animals in the first 90 days seem to be the most at risk animals. And also we seem to be seeing an increased number of people reporting the first case on their farm each year. Other thing we found that we need to investigate further is this possible link between cows on the farm as having teat necrosis, with cows on the farm with having udder cleft dermatitis and having dry or chapped teats in the milking herds. So at the moment we don't have a clear cause. So if we don't have a clear cause, it's very difficult to give clear evidence-based treatment for this disease. So we need to investigate this further. And at the moment, it doesn't seem to be that digital dermatitis is the cause, the main cause anymore. It seems like maybe it's a factor in some cases, but it isn't the etiology that we once thought it might be. So I just want to thank you for your attention, and I guess we'll be taking questions in a few minutes. Thank you, Mandy.
Thanks very much, Hayley. And yes, quite a conundrum there, but hopefully you'll be getting to the bottom of the causes of that. So I'd just like to thank all the speakers so far. We're going to head into our question and answer session, and my colleague Steve West is going to be helping on this. So Steve, I don't know if you're there. Well done, Steve. Thank you. So I'll hand over to you, Steve, if you'd like to start with the questions.
Well, thank you, Mandy. It's been a busy morning. I've been busily scribbling them down. What I'll do is I'll go back to the first group, if that's all right. Matteo and Stephen. The first question I've got for you, this was about Johne's and transmission of Johne's. Can it help if you pasteurize colostrum, and does that reduce the risk of Johne's transmission through the colostrum route?
I'm happy to answer that. Thanks for the question. I think the difficulty is we don't know for sure, but it makes perfect sense to reduce the pathogen burden for that particular for that particular donor. Personally, if one would use the pasteurization, I would personally use it still on a donor that is known negative, rather than on a positive cow that you then subsequently do the pasteurization on, because I think there is still the chance that you're not hitting them all, and therefore still keeping a background risk, even though reduced, of that transfer.
Perfect, thank you. Stay there, Stephen, because I'll hit you with another one while you're at it. Should I cull heifers born to a known positive dams, even if I've snatch calved them and fed donor colostrum? So this is based on the in utero transmission. You'd seen some of this. So is best practice really to look at the status of the dam and cull heifers accordingly?
It's a good question. I think it relates to the more long-term management of Johne's disease, which I think is really good and proactive. I think part of the dynamic is allowing these cows to calve down is that they are still sharing a dry period with negative cows, and therefore chancing that transmission of the known positives onto unknown positives, or hopefully negative ones. So therefore I think there's a benefit of not breeding these cows at all, because that will take that infection pressure out of the equation. That does, of course, depend on your infection pressure in the herd, as in if you've got 20% of your cows Johne's positive, you still need to keep on milking cows. So therefore, you probably want to sire them. You probably want to sire them to beef to reduce the chance of that infection spilling over to the next generation of dairy cows. So I think there's a benefit of selective breeding on these cows. I think more reversely, I think it is interesting that the data suggests that it is worth tagging the cow's offspring once the cow turns positive, to then think, okay, actually these cows born in the past are likely to become positive, and therefore treat them as red cows as well, even though test-wise they're not positives.
Marvelous. Matteo, I see you've come onto the screen. Did you have anything to add to that, or?
No, I think Stephen said everything. Yeah.
Lovely. Thank you. Thank you. So I'll move on to Erica and Javier, if that's all right, your colleagues there, RVC. So the question I've got there for you two is, can we clarify, just in practical terms, does the TB test affect the Johne's test results, or does the stress of TB testing affect the cow's immune response? So effectively, she knows better that she's definitely positive for Johne's disease. So do we know it's a factor of the TB test effect or the stress of the TB test effect?
I don't think we can really answer this, because what we have is just the JD test results at different points after the TB testing. I think biologically it makes sense that there is some type of interference, because these are similar pathogens. So it makes sense that there is something in terms of the reaction. But this is not something that we can confirm. We cannot confirm the mechanism from our study.
Marvelous. Okay, no, I understand. I know this is early, and you did speak quite a lot about your future plans, which I thought were really interesting. But have you seen from your initial your initial research, whether there was any variability with the effect due to age with cows, of the effect of TB on Johne's disease, or stage of lactation even?
Very good question. And actually, I don't think we split our animals based on age or stage of lactation. And part of the problem is that with age, there is already going to be some type of baseline effect of age on JD test results. I'm going to ask Erika, because she knows better than me. Erika, did we at some point look at whether there is differential effect based on age?
Yeah, we did include age in the model to see if it influences, like as a compounder, see if it changes the results. But in the model, it didn't. But the thing that does potentially have an effect is the stage of infection the animal is in. So if they're a green cow, not yet turned yellow or red, but they will at some point in the future be a red cow. At that stage of infection, their results may be actually quite similar to that of a non-infected cow. Instead, so there's like it's on a continuum scale, I guess, of how TB is changing their results, which makes it quite complicated. Yeah.
I think this is, thank you, Erika, because I think this is really relevant. We do believe there is an effect. It seems to be different if a cow is infected or not, but also different depending on the stage of infection at which the cow is, which is dependent on age. When we try to separate these things, age doesn't seem to be playing a role, but the stage of infection does. But they are all related, obviously, because as cows age, they are more likely to become closer to red, let's say.
Perfect. No, that's brilliant, and it looks like you've got plans with the extra funding to investigate some of this a bit further. So fantastic. What I might do now is I'll move across to James. I've got a couple of questions for James Breen. So this is referring to the Quarter Pro tool, the mastitis pattern tool.
Yeah.
What is the easiest way to record clinical mastitis cases? You explained it isn't always done. So what's the easiest way? And related to that, what denotes a clinical case? So is it something with clots? Is it something you feel you've got to treat? How would you define a clinical case for farmers?
Okay, yeah, thanks, Steve. I'll take the second one first then. So clinical mastitis refers to visible changes in the milk that you can see, or palpable changes in the udder that you can feel. So clots in the milk, flakes, bits, whatever you want to call it, is a clinical mastitis event. Now, I know within the industry there's a bit of a move at the moment to not treat these with antibiotic. There's various bits of guidance going around about the need, in inverted commas, to treat mild cases. Please do report them. If you decide not to treat with antibiotic, okay, that's another thing altogether. The fact of the matter is there were bits, clots, flakes in the milk. That is a clinical mastitis event. In terms of where to report it, then that's very producer dependent, isn't it? So the easiest thing perhaps is to report this to the milk recording organisation. So that's reported to the milk recorder. Some people don't wish to do that, and therefore the other way of doing it is to report it on farm yourself, either using on-farm software, Uniform Agri, Summit, GEA C21, Interherd. There's so many out there, but to have a database on farm that you report your mastitis events to, and or if you don't want to do it with software and you don't want to tell the milk recorder, well, then we've got it in a diary somewhere. And then it's incumbent on your veterinary advisor, your consultant, whatever, to end up taking pictures of your diary or scribing stuff out of the diary, because at some point it's got to live electronically if it's going to be of any use. And all we need is the date and the cow ID. Those are the only two things you need. You just need when that cow was detected with clinical mastitis, because then you can merge that data in with the milk recording file and relate that mastitis event to the calving event, and off you go.
Lovely. That's brilliant, James. Yeah, thank you. And I guess a whiteboard is also great because you can engage the whole team, can't you? So
yeah, but it's getting those clinical mastitis events off it, isn't it? They're there, but they're no good in isolation. They need to be in a database somewhere. Yeah.
Perfect. Thank you. Thank you. The next question, and Mandy, do shout at me if I'm taking up too much time, because I do want to go to Hayley in a moment. Is it important to record the quarter infected when we're recording clinical cases?
In terms of epidemiological patterns, dry period, lactation, season, then no. So quarters are not independent. So just because one quarter's got clinical signs now, it doesn't mean the other three aren't infected as well. And the reality is that it actually can be quite difficult for people to remember to record quarters. So Andrew will always tell you that cows have 5 quarters: front left, front right, back left, back right, and don't know. So actually stringing them together can be quite difficult. And you don't need quarter information for this. The cow, if you like, is the unit of infection. She has had clinical mastitis or not. So we need the date it happened to her. Less worried about the quarter. You can if you like, but it's less important in this sort of work.
Perfect. Thank you, James. And just a really quick one while you're at it. Where can you get access to this tool and how much does it cost?
It doesn't cost anything. It's freely available through the AHDB research partnership with University of Nottingham and QMMS. Go online, it's onto the Quarter Pro webpage. I'll put the link in there. You can share it on the chat, I'm sure. But click on the Quarter Pro webpage, and there's a nice little link there to the pattern tool and the 2 pieces of software you need. One software to convert your milk recording data. The other software is the pattern tool that will give you the actual pattern. Both freely available. You can both download them then.
Excellent, excellent, lovely. Thank you, James. We'll move across to Hayley then. Thank you, Hayley. It was a great presentation. I had quite a few questions here. The main one, the first one is: does necrosis, teat necrosis, always start at the base of the udder or at the teat end?
Yeah, it usually starts at the base of the udder or along the length of the teat. If it's at the teat end, it's probably something else, like black spot or something. But with teat necrosis, it's usually at the base of the udder or along the length of the teat.
I see, marvellous. So, well, related to that, how fast does it spread? How fast would that lesion grow?
It varies between cows, so that's something that it seems to be something that we need to do some longitudinal studies on, because I have farmers that report they'll see the lesion one milking, and the next lesion, at the next milking, the cow will have lost her teat. So it can be very rapid, and then others will say she's had it for a few weeks, not a problem, just bit sore when milking out. and then suddenly it'll progress, and suddenly she'll start licking, it'll get crusty, or it'll again, she'll start traumatizing it. So it seems to be very variable, but it can be very rapid. So that's something to be aware of, that because it can be so rapid, spotting these things early and trying to stop her from licking at her teats is crucial.
Excellent. And I've just got two more, Mandy, if that's okay. Is there any effect of pain relief on the heifer lactation 1 animal's reaction to this? And is that likely to sort of make it a safer place for people to work? And of course, how she reacts to licking and whatnot.
So I don't have any data to support anything around that. Obviously, it must be painful. It must be painful for these animals. But whether pain relief seems to stop the lesion progressing or not, I can't say. But it must be painful. So these animals really do deserve some pain relief.
Marvellous, lovely, thank you. And then the last question I'll ask you is, so can cows get reinfected? You said it was mostly lactation 1 animals. So are they effectively becoming immune, or is this something you want to investigate?
So this is something that we've been looking into, and we find that animals that do get these tend to be cold because of the lesion. About 60% of time, about 20% of times are cold due to these lesions. About 60% of these cases will have problems either due to them losing the teat and then go on to milk on 3, or due to mastitis. So a very large proportion of these animals will not make it to second lactation if they develop a lesion in the first lactation. So this is why it's so important to do more research. I have about 20% of animals that seem to recover. I've had a few farmers report that some of these animals will then come down with it in the subsequent lactation, but these are very low numbers because so few will report to me as them having been recovered. So yes, it is possible, but the biggest problem seems to be the first time they get the lesion and trying to deal with that to keep them in the milking herd.
Lovely. Thank you so much, Hayley, and thank you to all the speakers. I'll hand back to Mandy. Thank you.
Thanks very much, Steve. So we've just got a couple of minutes, and I'd just like to sum up. Thank you very, very much to all our speakers. I think we've outlined the power of data and just what data can do to help us prevent and control diseases on farms. It also gives us that insight into understanding how diseases can manifest in the herds. And really delighted that some of these projects have got future funding, and be really interested to hear the subsequent lessons that we learn from that. So I would like to thank all the speakers this morning very much. I'd like to thank the organisers and the people behind the webinar as well, who've made it all run exceedingly smoothly. Thanks to Steve for his questions, and thanks to you, the participants, for your very thought-provoking questions. There will be a short feedback survey which will appear on your screen once this webinar stops. If I could encourage you to fill that in, that will give us some feedback on how we can improve and take these webinars forward. The video will be sent to you within 24 hours of this, and slides will be available online. So thank you very much for joining us, and have a good afternoon.